CAR-T cell therapy. There are two types of T cells. CD4 expressing cell is a T helper cell, and CD8 protein expressing cell is a T killer cells. And T cells express the T cell receptor. And TCR can recognize the antigen. And this antigen, if it is expressed by cancer, then this can be cancer-specific antigens. But T cell receptor only can recognize the antigen which has complex together with the MHC1 protein. That's why if the cancer cell does not make MHC1, now this cancer cell cannot be recognized by T cell. This is one of the very common tumor evasion mechanism by downregulation of MHC1 expression. MHC protein is like ID card for our cells. So every cell in our body, they should express MHC, then they can be recognized as a self. It's a part of our own body. Now, if cancer choose not to express MHC, there is another police system, natural killer cells. Natural killer cell has a KIR receptor, and then if they recognize the MHC missing cells, then NK cell is activated, and then they can attack these MHC negative cells. KIR is NK inhibiting receptor. When NK cell contact different cell, and if they do not recognize the MHC complex, then they recognize this as a foreign cell, and they start to secrete chemical weapons. So in this case, the cancer cell can be attacked by NK cells. So originally, cancer cell had MHC, but now they removed the MHC, so they evaded the attack by T cell, but now they get attacked from NK cells. So to avoid this situation, maybe they can re-express MHC and then try to send out the messages, I'm part of you. So this kind of the hide and seek kind of a game. So they need to evade either from T cell attack or from NK cell attack. T cell's dilemma was the T cell receptor requires the MHC complexing with the antigen. So that's why the presence of MHC on the target cell is important for the success of recognition. To overcome these limitations, if the T cell receptor is changed by MHC independent receptor, then T cell may be able to recognize the target cell regardless of MHC's presence. Now, what can be that kind of the candidate of the receptor? Antibody is one example. Antibody recognition of the antigen does not require the MHC's complexing. So, if there is a way to replace the T cell receptor with antibody-like receptor and connect the T cell receptors, the signaling domain together, this new structure, so-called chimeric antigen receptor, so the abbreviated as a CAR, this new approach is called as CAR-T cell therapy. To apply this idea, from patient's blood, we can isolate T cells and then express that CAR, the chimeric antigen receptor, utilizing different gene vectors. And then the CAR-T cell express antibody-like receptor on the cell surface. And this can be multiplied and then put it back into the patient. Then these new T cells, these CAR-T cells, can attack cancer cell more efficiently than original T cells. The first generation of a CAR-T is composed of antibody-like binding motif connected together to T cell receptors signaling domain. And to make the structure simpler, the single-chain Fv as the small fragment of an antibody binding motif is utilized. To provide more functions, to make the cells survive better and proliferate better, co-stimulating domain is introduced. And next generation is incorporated this kind of a multiple co-stimulating domains. And also introduce the different kind of a functional motif to make the final therapeutic cell is more efficient. CAR T cell therapy is a very efficient approach, but also it has many challenges. First of all, when the cancer develops, they can make out new mutations and they make new clones of cancers. That's why even though one antigen can be targeted by CAR T, but there are a lot more different kind of heterogeneous antigen can be generated, then covering them all is very challenging. Also, overactivating of a T cell, it can generate large amount of the cytokine release, so-called cytokine storm can happen. So this can be very acute, the immune response to the body, which can generate the shock. That's why this symptom itself can be very dangerous to the patient. Also, so far, the CAR T cell is efficient against the single cell, such as blood cancer cells, but it has difficulties to penetrate inside big solid tumor, so there is some trafficking limitations. CAR is a chimeric antigen receptor, and first approach was modifying the T cells. That's why it was called as a CAR T. But same approach can be applied to different type of cells. If that CAR is introduced in NK cells, originally NK does not have specific binding motif. Now we can provide those specific binding ability to natural killer cells, then this can be called as a CAR NK. Same way, this approach can be applied to macrophages, then this can be used as the CAR-M approach. Originally, the CAR T was utilizing the antibody-like receptor, but maybe more expressed, different structure or more copies of a T cell receptor can be applied to T. So this is a modified CAR T approach. So currently, many different type of variations of a CAR approach is being developed for various cancer attack this strategy.